Hi all, welcome to my talk today. I'm a bioinformatics developer at Wolfram Alpha, and today I'm going to talk about biomolecules in Wolfram Language. So you can find the talk notebook that I am going to present today by scanning this QR code. So let's get started. So what are biomolecules? So biomolecules consists of different kind of molecules, primarily organic molecules that can be proteins, RNA and DNA, which are composed of nucleic acids, carbohydrates, and lipids. And today I will be primarily focusing on proteins, which are linear chains of amino acid that is in general folded into very specific shapes that is pretty predictably determined by their amino acid sequences. And the shapes determine the functions of the proteins. Proteins lies at the heart of almost every biological events in our body and in the living world. So that is, some of them are like structural support, biochemical catalysis, hormones, enzymes, and so on. So the basic structure of the amino acids are as follows. It has, all amino acids have a carboxyl group, an amine group, an alpha carbon, and an hydrogen atom, and an organic R group, and variation in the R group leads to different amino acids. These are the common 20 amino acids that is used to build protein structures in nature. This is created using molecule plot function in Wolfram language. These are some of the common nucleic acids that we find in nature. These are three common carbohydrates that we often come up with, that we often come across. Yeah, this is created using molecule plot 3D, and this is one example of a lipid, which is cholesterol, which we are all familiar with. So these three functions, biomolecule, biomolecule value, and biomolecule plot 3D was introduced in version 14.1, and this can be used to analyze and visualize biomolecular structures, in particular protein and nucleic acid structures. So I'll go into detail in the next few slides how it can be used. So biomolecule can be used to get the structure from different databases. So this is an example where I'm getting a structure of hemoglobin, which is used to transport oxygen to different parts of the body from lungs. We get the structure from RCSB protein data bank, which houses most of the experimentally determined protein and nucleic acid structures. And there is this unique 4-letter identifier that specifies a particular structure. So this is the identifier for hemoglobin. And you can see in the summary box, you will find different information of the biomolecule, like what kind of a biomolecule it is, how many chains are there, residues, and so on. So this is the structure of the biomolecule, which you can visualize using biomolecule 3D. This is obviously interactive. And as you can see, hemoglobin has 4 chains and there are 4 ligands. So now you can use biomolecule value to dig deep into how the ligands look and so on. That's what I have done here. So you can get the chain types of individual chains of the protein. So here you can see there are 12 chains, 4 are peptides, which are the protein chains, 4 are non-polymers, which are actually the ligands that you can see here, and 4 are water, which is not shown in the structure. This is like the default case here. You can get the explicit ligand as individual biomolecules using biomolecule value again, and you can visualize the heme ligand of hemoglobin, which is used to actually bind to oxygen and transport oxygen to different parts of the body. So this is a single heme ligand, and this is an iron atom that actually binds to the oxygen and carries it to different parts of the body. And you can see all the 4 ligands together using the show function. Now you can also visualize RNA, DNA, and protein nucleic acid complexes. So this is individual examples of that. So this is a pure RNA. And these are all experimental structures from protein data bank. This is a pure DNA, and this is a protein RNA complex. And for nucleic acids, you will see the purine and pyrimidine, individual purine and pyrimidine rings along with the backbone structure. So there are other different ways to visualize biomolecules. So the ribbon diagram that I just showed to you now is the most common, but you can also see just the backbones, different surface representations, as you can see here, individual atoms, the atomic, actually the molecular structure using ball and stick, tubes, and space filling. Yeah. So you can customize the chain and residue colors, which can be really useful to visualize specific chains, or when you are even teaching about protein structures to students. So this is a structure of human immunoglobulin G1, which is actually the most commonly found antibody in our blood. And antibodies are the molecules that body uses to fight all kinds of like bacteria and viruses and so on. It is also very important in like developing therapeutics. So this is the PDB structure ID for the antibody for the immunoglobulin G1, and I'm using this specific plot theme, and I have specified the colors for specific chain using color rules. So the interesting feature of the structure of antibody is it has 2 long and 2 short chains, and what I'm doing here is I have colored the long chains with shades of purple and short chains with shades of green. Yeah, so as I can say, this can be really useful to like teach students like, hey, this is the long chain and this is the short chain. This is like typical of most of the antibodies that we see in our body. You can also just highlight one specific chain using this kind of syntax and color rules. So here I am highlighting just the long chain A using red, and everything else is in gray. This can be done also in the context of highlighting residues. So this is a very nice example. So this is a ribonuclease inhibitor, which has leucine-rich repeats, which is a very common structural motif that is found in nature. And as you can see from the name, this is a lot of leucine residues. And what if I want to see where just the leucine residues are? So you can use the color rules like this, and you can just say highlight the leucine in cyan, everything else in gray. And as you can see in this structure, you can see where the leucine repeats are. And yeah, everything else is in gray. So another very interesting capability of the biomolecule, which makes it much more powerful, is you can fold a biosequence, a custom biosequence. This is done using the ESM fold API, which was developed a few years back by scientists at Meta. The sequence, the only restriction is the sequence must be a continuous peptide sequence within 400 residues, and this is set by the API itself. So here I've written a small code which randomly generates an amino acid sequence between length 1 and length 2. So here I generate the random sequence, which is 126 letters. You can get what the sequence is using the sequence string. You can get the biomolecule by just putting the biosequence itself as the input. And as you can see here, it says it is a predicted structure. It has a 126 residues as expected. And since this is a continuous sequence, it has just one chain, right? So now this is a predicted structure. There are 2 interesting things here. So the first one is you can see there are a lot of squiggly lines here, which is basically intrinsically disordered regions, in proteins, which is also commonly found in nature. And these are regions that does not have too many hydrogen bonding, so it lacks specific secondary structures. And this is expected because we are just randomly generating sequence. What is another surprising thing, and this is probably serendipitous, that we find so many helices, which might not happen for any randomly generated sequence that you like generate, right, using this code. So to show that, I have generated 5 more random sequences. As you can see, these are the 5 structures, and this has a lot more intrinsically disordered regions and lot less secondary structures, as you can see here. So let's try it out. Let's try to fold a random sequence between length 100 and 200. So yeah, and it does the job, right? It generates a new biomolecule, and as you can see how it is. So this can be very powerful in doing large-scale analysis using Wolfram language. So you can basically analyze the shapes and disordered regions that you find in the structures of generated from random sequence. You can compare them with natural proteins, or you can even fold like smallish sequences that you found from your own analysis or databases for whom the structures are not known. I will get to the questions at the end of the talk. I can see there are a few questions, so just bear with me for like, I think 20 more minutes or 10 more minutes. Thank you. So the two interesting applications that I could think of biomolecule that immediately comes to my mind is the one is protein contact map. So here what I'm doing is I'm extracting the backbone coordinates correspond. So there is one coordinate per residue, which is like representative coordinate that is used to generate the ribbon diagram using biomolecule value. And these are basically alpha carbons for amino acid residues and O3 prime for the nucleic acid residues. And this is like consistent with what is used in RCSB protein data bank for visualization. So here I actually get the structure, the biomolecule for an apolipoprotein, which is used in cholesterol synthesis or cholesterol pathway. And I get the backbone coordinates of this biomolecule using this function that is created using biomolecule value. You can just see like how the protein looks, like how the biomolecule, how the backbone coordinates are located. And this does have a very, very interesting structure, which I found really cool. So what you can do is you can create the distance matrix of this backbone coordinates, and thereby you can visualize it, and that basically is the protein contact map. So this is how the distance matrix looks, and it has a really like interesting structure. That's because I think obviously it's a pure geometric signature of the protein is coming from exactly how the backbone atoms are oriented. You can have a lot of information from here, like one being like how the off-diagonal elements or interchain like distances look like, and what is the overall structure. You can repeatedly use this function to explore different geometric signatures of different class or types of proteins. And you can also see from this this color, but this is in angstrom, that the kind of like the maximum distance between two residues or rough size of the protein is around 130 angstroms. And yeah, so you can see the actual biomolecule using BioMoleculePlot3D, and this is the structure of the protein. Yeah, another analysis that you can do is like just some intrinsically disordered regions. And I'm focusing on this particular term is because these are comparatively understudied things in protein structure, but recently it has been shown that intrinsically disordered regions are pretty important in many biological functions. But because these doesn't have specific secondary structures, often these are missed when you are trying to like experimentally determine the structure, or even when you are trying to predict structures. The sequences that correspond to IDRs actually have really low confidence. So often the structures that does have IDRs and that can be like seen using experimental methods often have many models. So typically a biomolecules have just one model, and by model I mean that like specific measurements of the same protein structure. But IDRs, because of its like intrinsic nature, have multiple models. So this is an example of a protein which has an intrinsically disordered regions, and you can see there are many models which is different measurements of the same protein structure. You can see using biomolecule value and model count that there are 10 models. You can also see how all the different models looks overlaid like on top of one another, and this is pretty fast. You can see like just to generate 10 structures, it takes less than 2 seconds here. And yeah, this is how this looks, right? So one might want to know like where is the maximum fluctuations in terms of residue location, and where it is like tightly packed and it's more like predictable, right, so to say. And as you can see here, you can just get the backbone atoms using the same function that I wrote using biomolecule value, and you can plot the fluctuations in terms of angstroms as a function of residues. And as kind of expected, at the ends it's really high because this corresponds to this end and this end, which is intrinsically disordered, and at the center where you have secondary structures, which is more predictable, you have really low fluctuations. This is another cool application that you can do with biomolecule value, but you can do a lot more. You can like think of like what is the frequency of amino acids that you find in specific classes of proteins, or how different they are, and so on. So another new thing that will be there in the next version 14.2 a service connection to ESM Atlas and RCSB protein data bank, which will allow you to really get much more tailored information from these databases. So ESM Atlas is a database that was published a few years ago that gives you access to over 770 million predicted structures. And that's a very big number because the total number of experimental structures in the protein data bank is roughly 230,000 at this moment. So, and ESM Atlas gives you access to metagenomic structures. So metagenome is basically the genome information that you find in bulk in nature, like on the surface of skin or in water bodies and so on. And these is relatively understudied part of protein structure, and it can give you a lot more intuition about what protein structures are capable of doing. So you establish a service connection using ServiceConnect. This is like if you want to fold a sequence, you give this input and you can give the sequence and it will generate the predicted structure here. The structure that is there in the database, you can give a unique magnify ID, which is again like the ID corresponding to the genome of a particular protein, and you can get the predicted structure using this command. This is just a given structure among many in the ESM Atlas. You can get the structure confidence prediction that can give you idea of how confident overall the structure prediction is. And actually in the ESM Atlas, they do tell how far removed it is from any natural known proteins that we know till now, or how close it is. And you can, a nice study can be just comparing the overall confidence to like how the distance between like known structures and unknown structures, right? And this structure. And so there are three elements to this. So the first one I would like to highlight is predicted local distance difference, which is PLDT. Is the measure of local confidence, which gives you an indication of how confident it is in measuring the atomic positions in a specific residue. And as you can see, it fluctuates overall in this protein. The second one being predicted aligned error. So this gives you an idea of how confident or how error prone the model is in predicting a position of a residue with respect to another residue. So this is a square matrix of the residue number times residue number dimension, and this is given in angstrom. So there is some regions with like high error, some regions with low error, and diagonal is obviously the residue is perfectly predicted with respect to its own position, right? So it's 0. Another one is being predicted template modeling score, or PTM, which gives you an overall idea of how the global confidence is. This is something that you can use for large scale analysis. Yeah, and this is how the structure looks again. So maybe like the low confidence area is somewhere around the end. Yeah, which is often like floppy and disordered. So you can also create a service connection to RCSB protein data bank. You can get the biomolecule using an external identifier. This is an example of a DNA protein complex. It's actually nucleosome, which is used for tight packing of DNA in our nucleus. You can get the entry data correspond to that protein. So this has a lot of interesting data, and you can do further analysis with specific elements of this data set. This is an interesting example. So you can do text search. So you can just give a plain text query and it will give you the relevant examples that you can get. By default, it gives 10 output, the PDB structure ID and so on, but you can customize it using specific parameters that I have not shown here. But it should be in the documentation. So I am looking for glucagon-like peptide 1. This is a pretty significant protein in context of like recent development in the pharma world. So GLP-1. This is the output that I get. This is the second structure that I get it as a part of the output. This is how it looks. And if I want specific information about the structure, I can use this entry data to get this. And as you can see, the detail is GLP-1 receptor bound to Pfizer's small molecule agonist. So recently, you might have heard in the news that like there's a big buzz around weight loss reduction drugs or drugs like Ozempic and so on. And these are drugs that can reduce the blood sugar levels in our body. And yeah, so this works by binding to the GLP-1 GLP-1 receptor and activating it to reduce the blood sugar in our body and also reduce hunger actually. And yeah, so you can also do sequence search, which basically is you can search for a specific sequence and see like how, like what structures you get. So what I'm doing is I am searching for a part of a sequence of human programmed cell death receptor, which is a very important receptor when it comes to cancer immunotherapy. And you see I get all this 10 different structures, and I can give a minimum overlap fraction. So I only want structures that at least have a 90% overlap with my input sequence. At least one of the chains of the structure should have 90% overlap. So I take a random structure from this 10, like structure 8. I see what the biosequence is using biomolecule value. I get the overlap, what the overlap is per chain, and using this WFR that I wrote this year. And you can see like for chain C, there is 98% overlap, and that's why it shows up. And if you see the structures, actually, this is the base structure that I have given. That's how it looks. And this is basically the structure, and there is maximum overlap with sequence chain C. And as you can see, this does look similar, right? So yeah, this can be really useful in searching for sequences. You can also search for specific structures, like structures that look similar to another structure. So you can give structure ID of like the PDB ID itself. So this is looking for another very important protein in cancer. So this is one of the most frequently mutated proteins in human when it comes to human cancer. And I'm trying to look for structure that is similar to this in this database. And as you can see, the 5 structures look pretty similar, right? Yeah, they might also be different studies made on the same sequence itself. You can also look for structures from similar to AlphaFold protein structure database. So this is another database that has over like 220 million structures, but these are structures in respect to standard organisms like humans and Escherichia coli and so on. So their structure ID is basically the UniProt ID, which is a unique protein identifier in UniProt database. So I'm giving the structure ID here. I am saying that please look up structures similar to this structure in the AlphaFold database, but give me experimentally determined structure from the protein data bank. There is shape match criteria, which can be relaxed or strict, which is based on different algorithms. And basically I get this 10 structures. I have these reference structures on my machine. And I also have the target structures, which I'm getting from this list, right? And as you can see, like, this is how the reference structure looks from AlphaFold protein structure database, and this is how the target structure looks. And I think what it does is at least one chain should look very similar, because proteins, obviously, experimentally determined proteins can have multiple chains. And as you can see, at least one of the chains indeed looks similar to what we are looking for from AlphaFold database. And AlphaFold database right now has only like single chain structures, to the best of my knowledge. So that's what you can look out for. You can also search by providing your own MMCI or PDB file, which makes it much more customized. So if you have your own structure file of proteins, you can give it as an input, and it will allow you to search for the structure. And I think the key there is file name. Yeah, and that will also be in the documentation. You can also do sequence motif search. So these are specific part of sequence or sequence fingerprint that is very important, that is seen again and again and again across different organisms. That means this has some specific functions, and this has survived the test of time through evolution, right? And I'm looking for now a very specific and famous sequence motif, which is called the zinc finger DNA binding motif. So what zinc finger does is basically it helps to stabilize the structure by forming bonds with specific residues. So these are the 10 structures that I get. And as you can see, this is the zinc finger part, and you can see check for other structures also. And here the score is 1 because I think the search is binary, either there is a zinc finger or not, right? Yeah. So this, I've come to the end of my talk. The future goals for the next year is to build more service connections, in particular to UniProt and AlphaFold protein structure database that I have mentioned, which will increase the access to biomolecules further by around, like, right now through ESM Atlas and PDB, we have around 800 million structures at our hand through Wolfram language, but this will increase it to like 1 billion structures, combined experimentally determined and computation structures. Add more features to BioMolecule-Plot3D to help it being more customizable for researchers and educators. Incorporate more protein structure analysis tools and incorporate more protein structure analysis tools in Wolfram language. I'd like to thank you for your time, and I'd like to thank my supervisor, Jason Biggs, and Wolfram Alpha Chemistry team for their help, feedback, and support in making the talk. If you have any questions, ideas, or suggestions, please email me at shrotiges@wolfram.com. And with this, I've come to the end of my talk, and I'm ready to answer your questions. Okay, so Matt has asked, can you read in individual genomic data and visualize it? Yeah, I mean, I guess by individual genomic data, you mean like sequences and all. So definitely there is biosequence in the language which can help you get the genomic data through a biosequence. And as long as it is within 400 residues, you can fold it and see the predicted structures. Also, it is possible to now, using the service connection, just input the sequence and see what structures are there itself in the protein data bank. And in future, once the service connections to UniProt is built, you can directly input the structures to UniProt and get much more information, because UniProt has relevant structure ID for both predicted and experimental structures. I hope like this answers your question. Dave has asked, can a 3D connection space mouse be used to rotate a biomolecule in Mathematica? If so, what Wolfram language functions are used to do this? I actually don't know what 3D connection space mouse is, but I will note this question and I will actually get back to you on, I will actually get back to you on this. So Bogdan is asking, can you compute molecule electrostatic potential or hydrophobic potential using Wolfram tools? It's a good question. I'm not aware of, I mean, I'm kind of fairly new to the language, so I'm not aware if there exists a tool like that. Maybe like other members of chemistry team, if they're there, can help me. Hydrophobic potential, there is I think WFR hydropathy plot that can actually give you the hydrophobicity of different residues when it comes to proteins, that I know of. And I think Jason Biggs has a talk today that talks about like, I think a different packlet. So we are trying to work on electrostatic potential, I believe, but I don't have a good answer to this question at this moment. What is the maximum size of a protein in number of maximum allowed atoms? So that's a very good question. So it can be pretty big, at least what I've seen in RCSB protein data bank. I don't know if there's a limit, there's a physiological limit that mathematically restricts it to be as large, but like there are viruses in the protein data bank that has, I don't know, like, say like the typical protein is hundreds of residues. A virus probably is like tens of thousands of residues at least. Yeah, so yeah, and then there are specific resources in RCSB protein data bank, the educational arm of them, which is called PDB11, where you can look for virus structures. Yeah, and they also often have icosahedral symmetry. So if you can look for that kind of symmetry in RCSB protein data bank, that can give you an idea of what are the limit for protein structures that are there in the protein data bank. Yeah. Any more questions? If not, then I would like to thank you for your time. Yeah, and if you have any more questions, feel free to email me at shrotiges@wolfram.com. Thank you.